So, a nice easy title here for this video, the endogenous pacemakers and exogenous zeitgebers. You might want to, for your notes, put EP and EZ for the remainder of it. But remember, in an exam you are going to have to write out these terms, so it might be nice to get used to writing out the full term. So, if you have a look on your specification, you need to know the effect of endogenous pacemakers, exogenous zeitgebers on the sleep-wake cycle. So that's what you're going to be asked about. How do endogenous pacemakers and exogenous zeitgebers affect the sleep-wake cycle? So we're going to have a look at the AO1 first. And key definitions, key terms are really, really important. So let's remember, we did go over this previously, but endogenous pacemakers are those internal body clocks, okay, that regulate all of our biological cycles. So things such as the effect of SCN, one single part of the brain, on the sleep-wake cycle, or the release of hormones, things that are inside us, parts of our brain, parts of our hormones. We've got exogenous pacemakers, so these are those external cues that affect our biological rhythms. So things such as daylight and how that affects our sleep-wake cycle. And it's really important to remember that EP and EZ, or endogenous pacemakers, exogenous zeitgebers, interact all of the time. Okay, they control and they fine-tune our biological rhythms. They don't work in separation. So this is a picture of your brain and the sunlight. So sunlight comes in via our eye through our optical nerve, and the two parts of the brain that we're going to be looking at are your pineal gland and the SCN. Don't worry about remembering the whole name of it. SCN will be absolutely fine. So, when we're talking about endogenous pacemakers, we're going to have a look at the SCN, animal studies on the SCN, and the role of the pineal gland in producing melatonin. So, the suprachiasmatic nucleus. Remember, you can just call it SCN in your exam. So, it's a tiny bundle of nerves found in our hypothalamus. And we've got one on the left hemisphere, one on the right hemisphere. And they are instrumental, they are vital in maintaining our circadian rhythm, so that sleep-wake cycle that occurs every single day. And it receives light, so we get light in through our optical nerve and it travels to the SCN within our brain. It works even when our eyes are closed. If we have our eyes shut, we can still detect the light coming in through our eyelids, and that message still goes to our SCN. And that allows us to adjust to daylight when we're asleep. So if we're sleeping, daylight can still get through our eyelids, through our optical nerve, to our SCN when we wake up. And this part of the brain is referred to as the master clock. Okay, it links to all other different brain regions that control sleep, arousal levels, and it helps control those biological clocks within our body. But there are a number of studies done on animals looking at the SCN. And the Corsi study in 2000, he destroyed, so completely terminated the SCN connections in the brains of 30 chipmunks, and then put them back into their natural habits. And what he found was that when he observed them over a period of 80 days, that their sleep-wake cycle had completely disappeared. The light now was going through their optical nerve, going to the SCN, but the SCN wasn't there to monitor and tell our bodies how to react to the light. So their sleep-wake cycles completely disappeared. And a significant number of these 30 poor chipmunks were killed by predators, most probably because they were awake when they should normally be asleep. So when it came around to night time and their SCN would have usually told their body to shut down and go to sleep, they were out having a great time. Of course, predators snap them up and they're dead. So there are more animal studies on the SCN rather than just terminating the SCN in a load of chipmunks. And Ralph, what he did was he bred 30 mutant hamsters with a far shorter sleep-wake cycle. So they made their sleep-wake cycle only 20 hours. And then he took the SCN cells from these mutant hamsters' brain and transplanted them into the brains of normal hamsters. And what he found was that the cycle of the normal hamsters defaulted to 20 hours. So when we took the brain cells from a mutant hamster who had a 20-hour sleep cycle and put it into a normal hamster, their sleep cycle reduced down to 20 hours. Okay, so both of those studies, both of these animal studies, highlight the important role that the SCN plays in establishing our sleep-wake cycle. So we're now going to have a look at the role of the pineal gland and melatonin. So the SCN gets information from the optical nerve when it detects daylight, and this passes this information onto something called the pineal gland. And during the night, this gland produces melatonin. And if you remember, melatonin induces sleep, so it makes us drowsy, makes us fall asleep. And it does this by inhibiting certain brain mechanisms. And this is one of the big causal factors of SAD melatonin. If you remember, in the last video, we said that melatonin being released too much in the morning is linked to seasonal affective disorder. And it demonstrates that there is a lot of interaction between these endogenous pacemakers, so melatonin being produced, and the exogenous zeitgebers. So we only produce melatonin as a result of exogenous zeitgebers such as light being detected. Okay, so it's this clear interaction between our internal mechanisms and these external mechanisms such as light. We've also got exogenous zeitgebers. We've got light and we've got social cues, and these can have a big impact on a range of different biological rhythms. So the first thing we're going to look at is the role of light and daylight. So this is one of the key zeitgebers in humans. Okay, it can reset the body's main pacemaker or master clock. It can reset our SCN. And it is clearly very important when we're talking about the sleep-wake cycle. And it also has an indirect influence on a load of other different body processes, such as hormone release. So depending on how much light there is and how much daylight, certain hormones will be either increased or decreased in their release. So there have been studies looking at the effect of light and daylight on the sleep-wake cycle. But an interesting study by Campbell and Murphy looked at whether we can detect light from other receptors in our body, not just the optical nerve. So what they did was they got 15 participants and they woke them at various times in the night using light shone on the back of their knees. Okay, so no light was coming through the optical nerve, just light on the back of their knees. And what they found was that this deviated people's and the subjects' normal sleep-wake cycles. Okay, 3 hours it changed it in some cases. So it shows us that light is exceptionally important in the sleep-wake cycle. And we don't even just rely on our eyes to influence the brain. If light is on a receptor on part of our body, then potentially that can influence our sleep-wake cycle as well. So we're going to go very quickly on how social cues can affect it. If you think about babies, for example, their initial sleep-wake cycle is exceptionally random. Babies don't want to sleep. They wake up, they're up whenever they want, they sleep whenever they want. However, they start to develop a circadian rhythm, a sleep-wake cycle, after about 16 weeks. And really, it's a question of why that happens. And the most likely reason is that it was imposed by parents that had a key influence here. So we give them, if I think about my 2 children, give them meal time at a certain time, bath time, bedtime. Okay, we impose this cycle every single night so the child learns when to fall asleep. And research also shows that adapting to local times for eating and sleeping is an effective way of beating jet lag. Okay, so there are certain social cues we can do, such as being a parent influencing a baby's sleep pattern, or changing how we eat or sleep in order to battle against jet lag. So there are social cues that can also affect our sleep-wake cycle. So now we're going to have a look at the AO3 for this topic. And firstly, we're going to look at the role of animal studies into the role of SCN. And there are some generalization issues here. Obviously, it's done on animals. It's very difficult to generalize it to humans. So you could have a paragraph on that in your essay. Also, there are ethical issues. If you look at the Corsi study, there was considerable harm and subsequent risk caused to those chipmunks. Okay, and that really brings into question whether the research should have been conducted at all. We've also got Siffre's cave study. So this does provide us evidence against the importance of exogenous zeitgebers, and it shows that light may not actually play an important role in the sleep-wake cycle. Because remember, his circadian rhythm, his sleep-wake cycle, was still about 24-25 hours, even though he wasn't exposed to any natural daylight. And it suggests that we do have a free-running circadian rhythm, and the only thing that influences this behaviour was his internal body clock. So it contradicts the role of exogenous pacemakers, okay, and that light has plays any effect at all on our sleep-wake cycle. There's also a number of methodological issues with these studies. Okay, much of the research was done on very, very small samples. If you look at Siffre's cave study or Ashcroft and Weaver's bunker study, that shows light might not have a major effect on the sleep-wake cycle, then they're only done on very small numbers, and it's impossible to generalize these findings to whole populations of people. It could be that there's individual differences in that research sample, that the participants responded in a certain way or an atypical way that other people wouldn't respond to. And Siffre noted that when he repeated his experiment back in his 60s, that his body responded very, very differently, and that his body clock actually took on more of a 48-hour cycle. So it might be that age actually has more of an effect than exogenous zeitgebers. There is a practical application to a lot of this research as well, and using light exposure to avoid jet lag would be one of those. Jet lag can be a really severe condition, can affect people for days and days and days. It can cause loss of sleep, loss of appetite, mood disturbances. By understanding the role of these exogenous zeitgebers and the effect that they have on our sleep, we can decrease the impacts of jet lag and the impact it has on our sleep-wake cycle. So, how do we actually avoid jet lag then? Any of you that go off travelling might be able to use some of this when you actually take big long journeys. I just found that exposure to light, or bright light especially, following an east-west flight, decreased the time, okay, that they suffered from jet lag. So if you're exposed to bright light, you had less jet lag on time of arrival. There were 3 groups of volunteers. He had one group who were exposed to continuous bright light, one group intermittent, so every so often bright light, and one group just to dim light. And what he found was that the participants who had the continuous bright light shifted their circadian rhythms back 2 hours, and that the intermittent group moved their sleep-wake cycle back 1.5 hours, and that the dim light group moved it back 0.6 hours. So what this tells us really is that the continuous group felt sleepier 2 hours earlier and woke up 2 hours later. So it was closer to the local time conditions to where they were flying. So it demonstrates that we can train our circadian rhythms, okay? So we can change our circadian rhythms to match certain time zones by using light, and that therefore reduces the impact of jet lag. Okay, so light and the practical application it has in reducing jet lag is really, really important. So one really important thing to remember is that this is an interactionist system. It is only in very exceptional circumstances that our endogenous pacemakers are going to be free running and not affected by any exogenous zeitgebers. So Siffre's case study, for example, it's an extremely rare actual scenario, and the whole study could really lack validity for that reason. It's very rare that we're going to be in a dark place for 2 months without any exposure to exogenous zeitgebers such as light. And in real life, the exogenous pacemakers and the exogenous zeitgebers do interact, and they should not be separated simply for the purpose of research.